Hi and welcome. My name is Natalie Caldell, and I have a short lesson for you today just to introduce the notion of abstraction. And the way I'd like to introduce it is to draw from some of the more mature engineering disciplines and talk about the idea of forward and reverse engineering, lessons we can learn from other engineering disciplines other than biological engineering. And then we'll try to apply some of that idea to the design of novel biological organisms. So my example of reverse engineering comes from the history of these gas cans that were used during the First and Second World War. If you're looking at the gas can that's on the left of the screen, that one looks really beat up as it is. It was used by British soldiers in the First World War. It was a terrible gas canister. It leaked terribly. They needed tools to open it, a wrench and a funnel, in order to get the gas to pour out, and the nickname it earned were flimsies. On the right side of the screen shows some different canisters. These are ones that the Germans had developed in preparation for the Second World War. I won't even try to pronounce that German name, what they called them, but the innovations that these gas canisters had, among them were the handles. You can see that there are actually three handles on the top, which made it easier for soldiers to carry between the two of them, easier to carry them if they were empty. You could hold two in a single hand. There was a snap cap now, so you didn't have to use a wrench and a funnel to get the gas out. There were changes to the way the sides were made, so there's an X on the side, which marks the side for strength, and expansion allowed to hold the gas better, had a plastic liner and pressed seams. So these no longer leaked. In fact, they worked so well that when the British and Americans saw these gas canisters, they kept them and they took them as examples to build ones just like them because they were such an improvement. The term that these gas canisters earned were jerry cans. I guess jerry was a nickname for Germans and Germany during the Second World War. So this is an example where somebody found something that worked extremely well, took that example, understood how it worked, and built something just like it. And that's a reverse engineering concept. I think it's funny, actually, if you look up jerry cans now, there's also a little bit of reverse historical engineering going on because you can find them online for sale. And if you'll notice in the middle of the screen, it says that it's an American icon. So the whole history of these cans as being developed by Germany is somewhat lost. But the lesson to take away from this notion of reverse engineering is that in one case, if you wanted to find the future, you can look to the past. So find good examples and make those the instances that you carry forward. Alternatively, what my mom might tell me is that you should just learn from your mistakes. If you see something that's not working, try to improve on it by finding things that work better and to make improvements that way. So that's an example of reverse engineering. And I just want to contrast that now with an example of forward engineering that comes from aviation. This is some schematics from the Boeing 777. What was special about this airplane is that it was designed completely on a computer. The ability to fly this plane and the ideas that underlie it came from what people already knew. There was about 100 years of manned flight under people's belt already, so they knew a little bit about what it takes to fly a plane. There was a good scientific understanding of the materials that were useful for flight, the physics of flight, changes in atmosphere, and how to react to them. There were already commercial airplanes, so the commercial needs of passengers, number that needed to be carried, how far they needed to be carried, all those were known entities. And there was consumer data, things about how much people were willing to pay for airline seats and things like that. And so to build the 777, a large team of engineers came together and assembled the components using tools, computer-aided design tools, and ultimately designed a very beautiful airplane which does in fact fly. Now, of course, nobody wants to get on an airplane that has never been tested, and has only been looked at on a computer. And the engineers involved in building the 777 did in fact do a lot of testing. They built some prototypes. There's a very interesting videotape that you can find on YouTube that looks at the stress on the wing. It's a wing test where they build a prototype of this 777. It was expected to be able to withstand a certain amount of deflection of the wings and the pressure. They tested at 150%. They spec'd the plane so that it could withstand 150% of any maximum wing stress that they expected. And so there's a very interesting moment in this video where the engineers are watching this plane get tested really for the first time and see it exceed the specifications. So I would encourage you to watch it. It's pretty short and very interesting. The takeaway from these lessons of forward and reverse engineering, though, are these: that to forward engineer a complex system like the 777, it required a notion called abstraction. It is a human invention. This is not something that nature gives us. This is a way that we as humans have devised to take a very complex system and engineer it. And so in this case of the 777, there were some engineers who were involved in system design, and there were other engineers who were involved in the electrical wiring, and yet other engineers who were responsible for putting all the pieces of those subcomponents together so that the electrical system wiring worked well with the system design. That those two groups may not have directly spoken to one another, but there were experts who could put them together so that they worked well together. And this notion of breaking a very complex effort down into smaller, separate efforts is this notion of abstraction. And some lessons that we can take away from abstraction, in this case, as you can imagine, everyone to build this 777 had to do their job right if this plane was going to fly. We rely on people within that abstraction hierarchy to do their job, and the people who integrate the job to do that properly in order for the product to work. There is no single person who could understand everything about the final product. There might be lead engineers in a particular effort as part of that abstraction, but there was no single person who knew everything about that plane and could be responsible for that. And then these integration engineers, the people who take the information from the different layers of the abstraction and put them together, those interfaces are critically important if the entire system is going to work well. Okay, so we've spent a fair bit of time talking about non-biological examples, and I'm going to end with the idea that we can apply abstraction to the building of very complex biological systems. And the way, this is one idea that we have for an abstraction hierarchy. It may be the right one, it may not be the right one, but it's the one that we're going to explore and try to work through. At the top layer of this abstraction hierarchy is the notion of a system, a complex biological system that would be built from subcomponents. Those subcomponents are things that we're going to call devices. Devices work in a particular fashion that we can describe. The devices themselves are built from parts. Parts would be proteins, RNAs, things that are actually doing the job inside the cell to carry out the intended function. And then, of course, those parts are encoded by DNA, G's, A's, T's, and C's, that will give rise to the elements that are the parts. So that's working from the top of the abstraction hierarchy down, working from the bottom up. We can say that sequences of DNA are what encode the parts, that assemblies of parts make up devices, and that assemblies of devices are what will make up a system. And so in a follow-up short presentation, we'll go through this abstraction hierarchy and actually apply it to biology and biological systems and look at what it gets you and why you might bother. There
Hey
is
dude,
an interesting
look who's here.
animation
It's
on
my
the
friend
BioBuilder
Izzy, the
website
eye jammer.
about...
She just stopped by for a visit.
Whoa! I never met a real eye jammer before.
Hey dude, great to meet you. I need to split some cells in the tissue culture room, but I thought you and Izzy could chat. Maybe she could tell you a bit about her project.
Awesome! So, what was your iGem project?
We tried to engineer a bacterial arsenic detector.
A what?
A bacterial cell that could tell us when arsenic is present in the water. See, arsenic contamination in drinking water is a big problem for people living in Bangladesh and West Bengal. And since there is no easy way to know if arsenic is in the water, people there have been drinking contaminated water and getting sick. So we wanted to engineer a biological system that people can use to test their water for arsenic.
I had no idea this was such a problem. What a great project to work on. How does your system work?
Well, our goal, or the system specification as engineers call it, was to have people mix a little bit of their drinking water with the culture of bacteria and know that there's arsenic contamination if the liquid changes color.
Awesome! But how did you get a bacteria to do that? I'm still trying to figure out how to make my bacteria balloon work.
Well, like all engineers, we use abstraction to help us. Let's take a look. We knew we needed to build a system that could take as input arsenic and produce as output a change in the color. So first, we compiled our system into devices. We needed one device that could detect arsenic and produce a signal. We called this our arsenic sensor and gave it a part number, BBA-J33201. And we needed a second device that could produce a color when it got the signal from our arsenic sensor that there's arsenic around. We called this color reporter BBA-J33202.
I don't get it. Why are you using two devices? Wouldn't it be easier to make just one, arsenic in and color out?
Well, you could do it that way, but then the only thing that the device could be used for is to report on arsenic contamination with color. By splitting the system into two devices, a sensor and a reporter, we hope future iGEM teams can use one or the other for new projects.
How?
Well, a team could connect the arsenic sensor device to a different reporter device that, say, produces light instead of color. Or a team could build a new system that detects, say, mercury contamination and then produces a color as a response. Yep, we design our system so that the people can more easily reuse and build on our work.
Oh, I get it. Maybe I could connect your arsenic sensor device to my balloon making device.
Exactly.
But shouldn't I know a little more about how the arsenic sensor works before I just hook it up to the balloon making device? Sally got kind of upset last time. I didn't stop the balloons from growing and growing.
That's why we enter both devices into the Registry of Standard Biological Parts. We also documented our project online at the iGene Wiki. The goal is that eventually, for each part and device in the registry, there will be a datasheet that summarizes all the relevant information about a device, so that you know how to use it in a higher level system, like the arsenic detector.
You mean just like this old data sheet I found for this 7404 hex inverter chip?
Yep, but I'm afraid there's still a lot of work to do before all the parts and devices in the registry have datasheets.
Great! Lots of work to go do. Let's get started.
All right, so there is lots of work to do, lots of good things to think about with abstraction. This is the end of the first abstraction lesson. As I say, in the second abstraction lesson, we'll go through the hierarchy and apply it to particular biological systems so you can see how it works. I hope this has been helpful and interesting for you, and hope to see you again in another BioBuilder lesson. Thanks.